General and special senses. The ear is associated with the detection of two special senses, equilibrium and hearing. The ear is divided into three anatomical regions: the external ear, the middle ear, and the internal ear. The external ear is the visible portion, and it collects and directs sound waves to the eardrum. The middle ear is a chamber located within the petrous portion of the temporal bone. Structures within the middle ear amplify sound waves and transmit them to the appropriate portion of the inner ear. The internal ear contains the sensory organs for hearing and equilibrium. The external ear includes the outer fleshy auricle, which surrounds the passageway called the external acoustic meatus. The auricle protects the opening passageway and provides directional sensitivity to the ear by directing sound inward toward the tympanic membrane or eardrum. The tympanic membrane is a thin, semi-transparent connective tissue sheet separating the external ear from the middle ear. The tympanic membrane is very delicate. The auricle and narrow external acoustic meatus protect the tympanic membrane from injury. The middle ear consists of the tympanic cavity, which consists of the auditory ossicles. The middle ear connects the tympanic membrane with the receptor complex of the internal ear. Also located in the middle ear is the auditory or eustachian tube. The auditory tube helps equalize the pressure in the middle ear with the external atmospheric pressure. The three auditory ossicles are the malleus, incus, and stapes. These bones are the smallest bones of the body. They connect the tympanic membrane with the receptor complex of the internal ear. These bones act as levers, transferring and amplifying sound vibrations from the tympanic membrane to the fluid-filled chamber within the internal ear. The sensory receptors for equilibrium and hearing are located within the internal ear. The receptors are found within the membranous labyrinth, which is filled with a fluid called endolymph. The bony labyrinth surrounds and protects the membranous labyrinth within a liquid called perilymph. The bony labyrinth is subdivided into three areas: the vestibule, the semicircular canals, and the cochlea. The vestibule and the semicircular canals form the vestibular complex. The cavity within the vestibule contains the utricle and the saccule. Hair cells within the utricle and saccule provide position and linear movement sensations. Hair cells within the semicircular canals are stimulated by the rotation of the head. The vestibular complex is part of the internal ear and provides equilibrium sensations by detecting rotation, gravity, and acceleration. It consists of the semicircular canals, the utricle, and the saccule. The anterior, posterior, and lateral semicircular canals are continuous with the vestibule. Each semicircular canal surrounds a semicircular duct. The anterior, posterior, and lateral semicircular ducts are continuous with the utricle. Each duct contains an ampulla that contains hair cells in a region called the ampullary crest. Each hair cell in the vestibule has 80 to 100 stereocilia and a single kinocilium. When external forces pushes against these processes, the distortion of the plasma membrane alters the rate in which the hair cells releases neurotransmitters. The receptors within each semicircular duct respond to one of these rotational movements. A rotation in the anterior-posterior plane, such as nodding yes, stimulates the hair cells of the anterior duct. A rotation in the horizontal plane, such as shaking your head no, stimulates the hair cells of the lateral semicircular duct. A rotation in the coronal plane, such as tilting your head from side to side, stimulates the hair cells of the posterior duct. The hair cells of the utricle and saccule are clustered in the oval maculae. The macula of the utricle is sensitive to changes in horizontal movement, whereas the macula of the saccule is sensitive to changes in vertical movement. Sensory fibers from the vestibular ganglia form the vestibular nerve. The vestibular nerve synapses on neurons within the vestibular nuclei, located at the boundary between the pons and the medulla oblongata. The two vestibular nuclei integrate and relay sensory information, as send commands to the other motor nuclei in the brainstem and spinal cord. The cochlear duct is located between two chambers filled with perilymph. The outer walls of the perilymph-filled chambers consist of dense bone, except at two small areas near the base of the cochlear spiral. These are the round window and the oval window. The round window is the more inferior of the two openings. It is a thin, membranous partition that separates the perilymph of the cochlear chambers from the air-filled chamber of the middle ear. The oval window is the more superior of the two openings. It is located within the cochlear wall, and the base of the stapes almost completely fills this oval window. The sensory receptors of the internal ear are called hair cells. Hair cells are highly specialized mechanoreceptors. They are sensitive to the movement of their stereocilia. The hair cells of the cochlear duct are located within the spiral organ or organ of Corti, located in the cochlea. This sensory structure rests on the basilar membrane, which separates the cochlear duct from the scala tympani. The hair cells of the spiral organ are arranged in two longitudinal rows, an inner row and an outer row. These hair cells contain stereocilia that make contact with the overlying tectoral membrane. The tectoral membrane is firmly attached to the inner wall of the cochlear duct. When a portion of the basilar membrane vibrates up and down, the stereocilia of the hair cells become bent. The frequency or pitch of a sound is determined by what part of the basilar membrane moves the most. Movement of the stapes sets up pressure waves within the perilymph. These waves distort the cochlear duct and the spiral organ, stimulating the hair cells. High frequency or high pitched sounds affect the basilar membrane near the oval window, whereas the lower frequency sounds affect the basilar membrane in areas that are further away from the oval window. The intensity or volume of the sound is determined by the amount of distortion in the basilar membrane. This is dependent on the amount of force applied to the oval window. This relationship provides a mechanism for detecting the intensity of a sound. Here are the steps in the production of auditory sensation. 1. Sound waves arrive at the tympanic membrane. 2. Movement of the tympanic membrane causes displacement of the auditory ossicles. 3. Movement of the stapes at the oval window establishes pressure waves in the perilymph of the scala vestibuli. 4. The pressure waves distort the basilar membrane on their way to the round window of the scala tympani. 5. Vibration of the basilar membrane causes hair cells to vibrate against the tectoral membrane, resulting in hair cell stimulation and neurotransmitter release. And 6. Information concerning frequency and intensity of stimulation is relayed to the central nervous system over the cochlear nerve. The auditory pathway is a 4-neuron pathway ending at the auditory cortex of the cerebrum. A large number of collateral fibers terminate in motor nuclei in the brainstem and initiate auditory reflexes of varying kinds.